Next speaker, Professor Markus Wenk, to talk about biological variabilities of blood metabolites. Professor Wenk is the Provost Chair, Professor, as well as the head of the Department of Biochemistry at NUS. He is also the founder. He established SLING, I guess not the alcoholic drink, but the Singapore Lipidomics Incubator, which is a interdisciplinary program at NUS dedicated to innovation, education, and partnership in lipidomics research. So without further ado, let us welcome Professor Wenk.
Yeah, well, thank you very much. I hope you can hear me. Chairman, can you just, great, and the slides you can see as well.
Yes.
Thanks for having me. Thanks everybody for tuning in. I'm not going to see anybody here, unfortunately. I'm in presentation mode, but I'm going to tell you about I'll give you an idea of what we do in these programs that were just mentioned. They're highly multidisciplinary as well. Move forward. So we study biological variability at the biochemical level because we believe it can tell us about commonalities among groups of people as well as individuals. Think of circadian control in human physiology, for example, where we share overall rhythmicity with some private patterns. Diet and absorption follows general pathways, but are highly individualized in efficiency and physiological response. So I'm going to tell you today about two types of variabilities. The one that is abbreviated here as CVG, and these are coefficients of variance for the aficionados, as opposed to the intra-individual variability. And we'll do this interactive. So think of yourself and how you change over time. That can be body weight, it can be physiological parameters as just introduced by Dino. It can be other measures. And think about health and potential onset of disease. I'm giving you one or two examples on the collaborative work with the National University Hospital. So it is not. Yeah. So it's exemplified really well in clinical practice when it comes to measurements of blood lipid levels. And we'll talk a lot about metabolites. For instance, the total cholesterol, triglycerides, and all the other things that you see there in black brackets, they're used by clinicians in order to make decisions whether somebody is in that box or outside the box, and whether to recommend a certain therapy or a change in lifestyle. Now, we are interested in a whole range of metabolites that are more in the research space, and we're asking ourselves, because they are increasingly being associated with certain physiological and pathological processes, whether we should use some of this biochemical information to guide clinical practice as well. So I'm going to ask you now to bring up your mobile devices and go to polyev.com/lipid and think of a particular lipid of interest. I've given you a few examples: the cholesterol, the fatty acid. Could be anything. If you cannot think of a lipid per se, then just think of a metabolite. We're close to lunch, and could be anything that you care about, that you might think is being absorbed by your body or has any function in your system. Give you a couple of seconds on this one, see if there's any response here. And while you do that, I think also keep that in mind, that particular molecule of interest. HDL, very good, thank you. Fatty acid. And so these are components that circulate right in our blood, and they nourish the system. They bring material from one organ to another. So I can see you're already medically oriented here. Thank you for this feedback. While others are coming in, we don't, for the sake of time, have the privilege to extend this. But please do keep the molecule that you're now reflecting on in the back of your head, because we will run a second exercise later on when it comes to how do these molecules vary within your body as opposed to your colleague sitting next to you or that you hang out with. Phosphatidylserine. Thank you very much. So we'll move on. And we use a range of technologies to do what you're going to see later on. At the core of it is mass spectrometry, but but we care a lot about, the demographics, the cohort design of the trials. How do we sample things? So there's a lot of not only basic science, but also increasingly engineering and how to keep the quality of our data up. And at the very downstream end, there's a group of colleagues in the teams now that are clearly data scientists that make sure we can get this data in an appropriate form to other collaborators. So a lot of clinically relevant work, and just a summary here, obviously not going to go through any of this in all detail, across the different clinical trials and spaces. A lot of diabetes and cardiovascular disease work, obviously, because of the interest in lipid biology, but also eye diseases. We've done quite a bit of work, chronobiology, now sports medicine is coming up with small trials. I'm going to highlight now, for just the scale of things here, associations of some of these molecules with type 2 diabetes incidence. And so this was one of the largest studies we've done a few years ago, where individuals were enrolled in this study. We've done baseline analysis of about 2,000 individuals, which at that time did not have diabetes, but followed them. They were followed over about a 10-year period, and some did then eventually develop diabetes. And so by looking at the metabolites measured using our technologies, you have an example here, we can then run associations between these novel markers and clinically established readouts for metabolic diseases such as BMI and insulin response. And so we found that's actually a balance of sphingolipid metabolism rather than just a bulk effect that runs the show here, and that specific details, fine chemical details, really make a difference. And maybe just to visually convey what we're getting at, this type of molecule we focus on here is really something that at the molecular level brings together fat metabolism, these are fatty acids here, amino acid metabolism, that's the anchor between the fat component, and finally the sugar head. So it brings three macronutrients floating around in the body into a molecular entity that we can follow, and of which there are many. Because we can do these things at scale, it offers other opportunities. We can do genetic associations. Other groups at NUS are interested in exploring this data for nutritional reasons. Because these are taken up, you can make associations between the diet and some of these floating molecules. Now, to get back to this variability, I'm going to introduce a smaller scale study that is currently ongoing, and we are very excited about the longitudinal aspects of that. In this particular case, a small group of middle-aged individuals, community dwelling, were recruited by the cardiovascular team. So they were interested in taking individuals that had a varying degree of risk for atherosclerosis, from low to maybe medium, but they were all subclinical. So none of them really had full-fledged heart issues. They were followed for 6 months, and they were then sampled, blood was taken at these time points, but they've also been undergoing detailed imaging of the coronary arteries. And so we had not only the biochemical data, but also the image information of these. And you can then run associations, and this is done here against risk scores that are very well established in the clinic, such as the Framingham risk score, where you can look into the different types of plaques that you can image, either lipid-rich or calcified or fibrotic, and see how well do they really associate with these clinical measures. And the association is moderate. Now, this is the data of the biochemistry, and I'm showing this to you here as a loop movie. So, just to go back to the abbreviations, this is not all too interesting or maybe relevant for today. The TG is this triglyceride. So this is a lot of fatty acid in this thing. And there are other molecules here, but it doesn't matter for now what they are. Look at the variation. These are the 6 months of a single individual in a loop. So I should say that. So it loops back the movie. But it gives you an idea that these molecules, they vary over the time span of 6 months. in a particular individual. The red line is the sum composition in concentration values for all of these molecules. So we can follow them individually quite well. You see some swing a bit more. So have a look at this pattern, and now think back to the compound of interest that you submitted earlier and ask yourself, how would that vary? This is a particular volunteer. If I show you the same for another one, it would vary as well, but it would not vary precisely the same. So there is a high degree of individuality to how these things actually fluctuate. So let's go back to poly-V and bring back your molecule of interest and guesstimate. You know, if you had to put down on a range in percentages, technical variability, I can already give you an idea that's hopefully going to be low. And then how would that molecule vary within you as opposed to individuals? Somebody of your same age, could be a different gender, maybe then that would affect that as well. But give some form of range here. CV is a form of relative standard deviation. Give you a few seconds. This is really difficult to do in the short time frame, but I'd like to run this exercise anyway. CVA, typically we clamp at about 10 to 20%. So that gives you an idea that, you know, anything biological hopefully is bigger, because if it's not bigger, then we're not able to use our technologies to interrogate physiology. So hopefully the CVG and the CVI are in the range above the technical variability. Now, the question that I'm trying to get at is, do you think you vary more within yourself when it comes to these fluctuations of molecules, but it could also be body height, of course, and weight, or do these parameters vary more between individuals? Okay, I'll give you a couple of extra seconds. We've got 10 hits, then we've got to move on for the sake of time. Yeah, usually it crystallizes quite well already. So there is maybe if I can interpret this noisy data, there is a tendency to an agreement somehow that maybe the CVG, the group individuality between individuals, is maybe a bit larger than the variability within us. Thank you for this feedback, and I'm going to show you the data now. And indeed, you are absolutely right. So we can explore now using our technologies, which is high density across these different chemical compounds, and look into between subject variability shown here as the solid bars, as opposed to the within subject, within us over time variability shown as the lollipops. And you can see it's really varied. It's very distinct for certain molecules. For overall, the between subject variability is larger, but we do have examples where it's vastly different, and we are paying a lot of attention to exactly these differences. Now, why does this matter? And this is the same data shown in a slightly different form to represent the abundance of these molecules, but you see the data population on this correlation chart is really skewed towards the top. So there is typically more variability within, between us as opposed to within us for a given compound, in particular for these fatty acid based triglycerides that are of interest in the clinic. Now, we then used exactly these data that varies more between us and check, well, you know, how does that really associate with these risk scores? And we found that associates fairly well. And therefore, you know, you could use a single or a measurement, or maybe two measurements over 6 months to get a good idea of your risk there. Now, when it comes to those compounds that vary more within us, we really pulled this to the individual level. So we looked into the variability of these compounds in each individual over the 6 months. And we found that, you know, some of them really associate, this is the variability of an individual compound with your sort of a score for the plaques, in this case lipid-rich plaque. So you see we have an association of certain LPCs, whatever they are, with lipid-rich molecules, and make a point that maybe these could be candidate markers to get a sort of a risk assessment for somebody that is subclinical for atherosclerosis that may in the future have problems with this presentation. So this is a summary here. I have no time to go through all of this, but we have now a reference data set for variability of these compounds, research grade compounds in small populations. We're using this to understand what is the value of that based on the ratio between individual as opposed to within individual variability for sampling. How often do we need to measure an individual before we hit the value on the spot? And we can use these lysolipid compounds in association with unstable lipid plaques, potentially also in the clinical use in the future. Now I'm going to give you a short introduction to a program that really takes some of these analytical sciences fairly deep into the clinical hospital space at NUH. It's a tripartite partnership between NUS, an industry partner, and NUH. I'm going to actually bring up a short video for a couple of minutes that we can go through. Then you have a different voice and get a couple of visuals as well.
This hub is quite unique in really two major elements: its downstream reach to capture value and its level of partnership.
The hub and satellite model allows research to be conducted centrally but shared broadly through key partnerships and alliances supported by Agilent, the university, and the government of Singapore.
From our earliest discussions developing this iconic hub and its focus on translation and capture, we also envisioned that it could be a partnership model that would be replicated in other areas, a model for how research is advanced and translated into real use, a model that could be applied in additional areas of healthcare, as well as in other fields such as maybe nutrition or energy.
So there are three elements here, maybe. Number one, we plan to build a one-stop hub for clinical testing. Two, we plan to translate first to market mass spectrometry-based technologies. And three, we hope that this hub actually serves as a model for replication in other areas, such as nutrition, for example. So I think it is actually first of its kind and truly integrated from A to Z.
The aim of the Blue Node is to develop innovative analytical tools for the detection and quantification of small molecules in biofluids, tissues, and cells. From an analytical point of view, its activity aims at developing new methods for the separation and the high throughput detection of metabolites by mass spectrometry.
In the data integration node, we are interested in streamlining the workflow to curate, store, and analyze the massive amount of data that are generated from different hubs. We are ultimately interested in building predictive models for chronic diseases such as type 2 diabetes and heart diseases based on data collected from the local population.
The purpose of this node is to really narrow down all of those components and find the specific ones that help doctors make decisions for their patients. Blood tests are one of the basic ways that doctors monitor the health of their patients. But between health and disease, many things change with the blood.
The capture node aims to translate both existing and new lab tests to the mass spec platform, thus providing better quality for better clinical diagnostics. The core competencies represented by our 4 nodes have, until now, been acting in silo. And through this collaborative project now, we are breaking down these artificial barriers.
This collaboration allows NUH to participate in emerging technologies and test development. Exciting new research results from the R&D nodes suggesting new biomarkers can be test-fitted through the integration and translational nodes, and thus validated and tested for utility?
Well, the initial focus areas are clearly cardiovascular diseases and diabetes. These are currently focal areas in healthcare in Singapore and globally, of course. So Singapore is a place for innovation. Stop this here. Quickly go back to my last slide and end there, I think, with this one. So that's in essence that hub that was mentioned. There's a research lab that is in the NUS Medicine Science campus of NUS, but it's closely connected and also in physical proximity to the School of Public Health. We work very closely with some teams there. We've got some satellites also in the medicine space, the cardiovascular research program, for example, but then also really NUH. So it's a corridor physically and team-wise that brings together individuals that can either take a project from a research lab and ask, can we translate this eventually to the clinic? DLM stands for the Department of Laboratory Medicine. EMD stands for Emergency Medicine Department. So these are already part of NUH, right? But it could also go the other way around, and we've learned that during COVID that's maybe a maybe even more interesting way of looking at the challenges by stratifying, you know, the material as it comes into a clinical arm that is operations, but also a research component. We might in the end be able to come back and ask questions on utility in very different ways. So this is modular, and we are very, as you've seen, very multidisciplinary, got a broad team, broad student base, and love to take any questions and maybe even have a discussion with you if you're interested offline. Thanks for having me again.
Thanks, Markus.